Welcome everyone, Dr. Linda here. In this first short video, I'd like to talk about the neuron. Now, when we talk about neurology, there are two types of cells. There's the neuron, which is the only one that's electrically excitable. And then you have other cells called glial cells or neuroglial cells. Let's see if I can spell that for you here. Let's put it out here. Neuroglial. And I've seen this pronounced neuroglial. I've seen it pronounced as neuroglial. It's two words combined, and it really means nerve glue. And you'll learn about them predominantly in lecture. You'll hear of neuroglial cells that are called Schwann cells and oligodendrocytes and astrocytes and ependymal cells. So you're going to hear those discussed a little bit more in lecture. I may mention one or two neuroglial cells in the lab portion as it's relevant. But for the most part, you're going to be seeing the anatomy of a neuron on the lab. And when we look at this, this should look a little bit familiar. When we covered tissues, remember in the very, very beginning of the term, we spoke about epithelial tissue, connective tissue, muscle tissue, and nerve tissue. And we saw something like this when we spoke about nerve tissue. The neuron has a few moving parts to it, right? It has these branches that we see here, which are called dendrites. These are all dendrites, and it comes from the Greek word dendra, which means branches or branches of a tree. Then we have a cell body, this portion. And sometimes the cell body is called a soma. So those two terms are interchangeable, cell body and soma. And then we have this long cylindrical structure that is referred to as the axon. And information moves in the direction of this arrow, right? So information comes in to the dendrites. The cell body is going to process that information. And there is this little dotted section, we could see it here, that's called the axon hillock, which is the dilated part of where the axon is, the axon hillock. Then we have the axon. And at the very end is the axon terminal. And this little section here looks like it's boxed in, but this should look familiar because this could be the neuromuscular junction, right? Where the axon terminal butts up against something else. In this case, it's butting up against another cell, but it could be a muscle cell. So at the very end, at the neuromuscular junction, what do we have stored in those synaptic end bulbs are neurotransmitters. And if it was the neuromuscular junction, then that neurotransmitter, acetylcholine or ACH, should sound a little bit familiar. Okay, so let's take a look at the model and see what some of these look like on the actual model. Now, this part right here on the top, right there, that would be attached to, let's see if I could do it, to right there. All right, but to make it fit on this small board, they cut it. But this really would be an extension of right here. So we could see some of the main structures. I'll circle them. The dendrites. Here's the soma, or the cell body. Right in the center, we see the nucleus, with the nucleolus. Here's the axon. Here's the axon hillock. And just after, right, so here's the dilated part, the axon hillock, the hill. Just after it is what they refer to as the initial segment. And the initial segment right here is where sometimes they also call it the trigger zone, which is where depolarization takes place, or where the action potential is initiated, right at that initial segment. Okay. Right here, this dilated part, here and here and here, those are the axon terminals that we mentioned earlier. Now, what do we find at those axon terminals? We find neurotransmitters. And it's really these neurotransmitters that end up being the key messengers of whether we're going to have something excitatory that's turned on, or we're going to have something inhibitory that's turned down. It really depends on the neurotransmitters, because some neurotransmitters are excitatory and some neurotransmitters are inhibitory. I'll give you an example. We have a neurotransmitter that is called GABA, G-A-B-B-A, GABA. We have another neurotransmitter that's called glutamate. Glutamate. Now, glutamate is an excitatory neurotransmitter. It makes your brain somewhat stimulated. When you get up in the morning, for example, and your alarm goes off in the morning, you should be jumping out of bed, right? Boom, that's glutamate. That's sparked up high. That should help you get out of bed, get dressed, do everything you need to do. Glutamate gets converted by evening time into GABA. And GABA is more inhibitory. As the sun goes down, the flower closes up. So glutamate is more stimulatory, it's the on switch, and GABA is more inhibitory, it's more of the off switch. So as you go to bed late at night, you should be a little bit tired. That's because GABA is kind of turning things down and getting you more relaxed. Okay, so some are stimulatory, some neurotransmitters are inhibitory. Now, there's some people that get up in the morning, and when they get up in the morning, the alarm goes off and they hit the snooze button, or give me 5 more minutes, or just 8 more minutes. Oh my gosh. That means that they're lacking glutamate, that perhaps they're GABA dominant. Okay. When you go to bed at night, you should be nice and calm and relaxed. If your brain is too stimulated, then you're flip-flopped, right? Then your glutamate is dominant at night when it should be dominant in the morning. All right. So these neurotransmitters are very telling, they're very strong, and they do control a lot of our physiology. Okay, similar structures here. I just want to point out on the left-hand side, there is a neuroglial cell that's referred to as a Schwann cell. Now, Schwann cells produce this. It's called myelin. And myelin is all of this stuff that we see out here, all of this white insulation that we see on both sides here, that is myelin, or the myelin sheath. Now, what myelin does, myelin is produced, if we're talking about the peripheral neural system, the PNS, it's the Schwann cell that produces myelin. If it's in the central neural system, which is the brain and the spinal cord, then there's a different cell that myelinates. And the one that myelinates, I'm going to put oligo. there's a cell called an oligodendrocyte. An oligodendrocyte. So an oligodendrocyte is also going to produce myelin. However, it's a type of cell that myelinates only in the central neural system, which is the brain and the spinal cord. Your Schwann cells produce myelin in the peripheral neural system, which is everything outside of the brain and outside of the spinal cord. So myelin is an insulator. It's made up of fat, it's made up of cholesterol, and it insulates the wiring. Okay, think of plugging in your smartphone. You don't see the actual wires, the electrical wires. You see this insulation. It's usually a white coating or a black coating around the wires. That would be equivalent to myelin. It speeds up the conduction of impulses. It speeds up the conduction of action potentials. Without myelin, you get misfiring of electrical information. And there's a disease that we know of called MS. And MS stands for multiple sclerosis. Multiple sclerosis is when the individual has an autoimmune condition against either their Schwann cells or their oligodendrocytes. And it's diminishing and shutting off the body's ability to produce that fat and cholesterol that produces the myelin or that insulation. Okay. Let me show you. So right here, this happens to be a Schwann cell. That's one Schwann cell. Up here is another Schwann cell. And then down here is another Schwann cell. And between Schwann cells, I'll put it in a square here, it's called node of Ranvier. The node of Ranvier happens to be that junction or that union of where two separate Schwann cells come together. Okay, and the node of Ranvier is where these electrical impulses jump or leap. They will jump over one node to the next Schwann cell. Okay, and when you learn about different types of conduction in lecture, you will learn about saltatory conduction, which comes from, in Latin, saltar or saltare, which means to leap or to jump. Saltatory conduction. The electrical impulses jump from node of Ranvier to the next node of Ranvier. It's what allows the action potentials to be so fast and so quick. Okay. Same type of structure, just a different type of model, but again, showing the same structures where we see the dendrites, which are these branches that we see here on the outside. We see the cell body or the soma. We see the nucleus in the center. We have an axon hillock, which is the dilated part right here. And just distal to it, again, is the initial segment. Some call it the trigger zone. Here's your axon, and here's another Schwann cell. What does the Schwann cell produce? It produces the myelin sheath, which is, again, made up of fat and cholesterol and insulation to speed up the conduction of impulses so that it travels super quick in that direction. Close-up view, the orange structures that we see here, the orange ones are the axon terminals. Remember what's stored in those axon terminals, those synaptic end bulbs, you got neurotransmitters. But the yellow structures are the actual dendrites. Here's the soma or the cell body. Again, the nucleus with the nucleolus right in the center. Here's the axon hillock and the initial segment. Here is this entire structure right here. That's called the Schwann cell. And again, it produces that myelin. Here is the nucleus of the Schwann cell, right there. That's the nucleus of a Schwann cell. And between each Schwann cell is going to be the node of Ranvier, the node of Ranvier. Okay, let's just see. There's nothing here that I'm going to have you identify. I'm not a big fan of these terms, so don't worry about those. There is a structure right here that's important called an endoneurium. That's that gray membrane here called an endoneurium. Just like when you covered muscles, we learned about an endomysium, a perimysium, and an epimysium. We have these similar layers with the nerve where there's an endoneurium, a perineurium, and an epineurium. But the innermost is called the endoneurium. And again, you can see the endoneurium down here at the bottom, that gray section right here. Okay.